Hi class, let's start our discussion on the cells. You can notice that, as we talked about earlier, that the cell is the basic structural and functional unit of life. All living things are made up of cells, and that is something that I need to hammer home, as I've already been doing for you every class period that we've been meeting so far. The cell is the basic unit of life, which means that every living thing is made of cells. If that thing that you're looking at is alive, by definition, it has to have lots of cells in it. There's also cell theory. Cell theory states that every new cell arises from a pre-existing cell. Now, of course, if any of you like to do philosophy or think a little bit outside the box, this cell theory begs the question, well, what about the first cell? Where did that first cell come from? Cell theory does not address that question at all. That is a completely different conversation, a completely different discussion, which has very different ramifications and is much less, or is not as well established. I should leave it at that. Moving on. Why are cells so stinking small? Why on earth can't cells be ginormous? Why can't they be large? Have you ever watched that 1950s classic, or maybe it's 1960s classic, The Blob, where you see this giant single cellular organism the size of a building roam through a town and swallow people in cars? Why does that never happen? Why do we never have killer amoebas that swallow people whole? The reason for that is that cells need to transport nutrients via diffusion through their cytoplasm, through the inside of the cell. And if the cell is very, very large, it takes a long time for those nutrients to diffuse into the center of the cell and for the waste products to diffuse out of the cell in a timely fashion. There is a time scale, or there's a time limit, I should say, that needs to be imposed on the chemical reactions that keep the cells alive. And if we don't have those chemical reactions taking place in a timely fashion, the cell will die. So we can have a small cell because that small cell has a very large surface area to volume ratio, so that we can have these chemicals diffuse in and waste products diffuse out within a timely fashion. So let's look at some hypothetical cells here. Yeah, I got a little tongue-tied here. I should finish my coffee. So we have one cube that's 4 centimeters by 4 centimeters. So this would be a cuboidal cell. So if we look at this cube, its total surface area is 96. Oh, that is a typo if I ever saw it. That should be centimeters squared, not centimeters cubed. Ooh, shame on the publisher. So we should be total surface area of 96 square centimeters. And if we look at the total volume is 64 cubic centimeters right there. So 96, if we look at our surface area to volume ratio, the surface area to volume ratio is about 1.5 to 1. So the surface area is about 50% larger than the total volume of the cell. If we looked at the numbers involved and we disregarded the units. Now if we looked at these 2 centimeters square cells, cubes, each individual cube has a smaller surface area than this overall cube, but because there's more of them, that same amount of stuff is going to fit into more cells that each have a combined greater surface area. So we go from 96 square centimeters for the surface area to 192 square centimeters, even though the overall volumes are going to remain the same every single time. So we can find that 192 divided by 64 gives us a 3-to-1 surface area to volume ratio. And then if we go with 1 centimeter square cubes, the total surface area is going to be 384 square, not cubed, square centimeters. And 384 divided by 64 gives us a 6-to-1 surface area to volume ratio. So if we looked here, this large cube takes a very long time for nutrients to diffuse in, but these smaller cubes can have nutrients diffuse all the way to the center much quicker, in a much more timely fashion. How do we study cells? Cells typically are going to be studied through multiple methods. Some are going to be chemical assays. Other methods are just going to be looking at the cell. To look at a cell, we typically just use a microscope, which is a tool that we use to look at very small things. Scope I believe, is a root word for C. Micro is definitely a root word that means small. So we can use a light microscope. Light microscopes are very common at undergraduate and high school levels. These are the bread and butter, so to speak. These are the workhorse of the undergraduate laboratory experience. These compound light microscopes use low magnification. When I say low magnification, I mean less than 1,000 times magnification. But generally speaking, when we look at the magnification of a light microscope, we're going to focus on anything between 40 times magnification, and 400 times magnification. Very rarely do we use that 1,000 times magnification, because most undergraduate light microscopes just don't have the optics to use 1000x very effectively, but they can if they have to. These compound light microscopes are going to use visible light beams. Sometimes they will use a mirror to reflect sunlight. Other times they'll just have an electric light source. And we can also even view live specimens with these compound light microscopes, which makes them very effective for environmental science experiments. We also have a transmission electron microscope. Transmission electron microscopes, TEMs, are going to produce a two-dimensional image, and we use a beam of electrons. Instead of a beam of visible light photons, we use a beam of electrons to view the internal structure of a cell. Because electrons have a much lower wavelength, or much higher frequency than visible light photons, we're capable of seeing much smaller things in the cell. So we can have very high magnification and can have very high resolution. A downside is that since we're using electrons as the photo source, we need to take the picture in complete vacuum or near vacuum, because the air molecules would move the electrons around and screw up the image. And the process of sucking out all the air and making that complete vacuum or near vacuum means that it kills any living specimen by looking at it in a transmission electron microscope. We also have scanning electron microscopes. Scanning electron microscopes are going to be very similar to transmission electron microscopes. The big difference is transmission electron microscopes. These transmission scopes are going to blast the structure with electrons and have the electrons go through the sample, whereas the scanning electron microscope is going to hit the surface of the sample with electrons and have the electrons bounce off the surface. So because they are bouncing off the surface of the tissue, we can produce a 3D image of the tissue through a process called Z-stacking, which is beyond the scope of this course. We can then have very high magnification. Again, because of the complete or near vacuum, we have no live specimens. Here are some blood cells that are viewed through different microscope sources. So we have a traditional light microscope that we have for this blood vessel, and this is at 250 times total magnification. So this is right in the sweet spot for a visible light microscope in terms of overall magnification. Light microscopes work very, very well at this range of magnification. Here we have a transmission electron microscope, and this transmission electron microscope is at 10,000 times total magnification. So that itty-bitty tiny little red blood cell that I'm pointing to right here is now ginormous on the screen. But this only is going to give us a two-dimensional view. And then here we can have a scanning electron microscope, and the magnification in this image right here is about 250 times total magnification. So about the same as the light microscope over here on the left. But if we notice on the right side of the screen, since we used a scanning electron microscope, we have much more detail, much higher resolution at the same magnification compared to the light microscope. Scanning electron microscopes, generally speaking, are the gold standard for microscopy, and a medium quality scanning electron microscope, so one that you can do some pretty good work with and that it's fairly reliable, is about $1.5 million to give you a price scale. Whereas a compound light microscope, a medium quality compound microscope, is about $1,500 to $2,000. So these scanning electron microscopes are about 1,000 times more expensive than the compound light microscopes, which is a big reason why we don't use them in undergraduate laboratory settings. So, class, concept checked. What is the most basic unit of life? Is it A, atom, B, molecule, C, cell, D, organ, or E, organism? Go ahead and pause the video, rewind your notes, or flip back in your notes, check your textbook, or rewind the video and get me an answer. 1, 2, 3, 4, 5. The correct answer is C, cell. The cell is the most basic unit of life. Let's keep moving on. There are two major kinds of cells that are in all living organisms. We have cells that do not have a nuclei and cells that do have a nuclei. Prokaryotic cells are cells that do not have a nucleus. That's a key characteristic, key hallmark. And there's two domains that fall into the prokaryotic category: domain bacteria and domain archaea. And then there's eukaryotic cells. Eukaryotic cells are cells that have a nucleus. And eukaryotic cells have a domain all to themselves, in that they're all the cells that have a nucleus. Since prokaryotic cells do not have a nuclei, we think that they're typically simpler cells. And since they're simpler cells, it's believed that they are the first cells to arrive on the scene. Eukaryotic cells, since they have a nuclei and many membrane-bound organelles, are more complicated and thought to have arrived a little bit after the prokaryotic cells. But this is actually slightly out of date. More current theory is showing that both prokaryotic and eukaryotic cells arrived at approximately the same time from a common ancestor. But that's okay. So let's look at prokaryotic versus eukaryotic cells. What do they have in common? And then what do they have different from each other? All cells, regardless if they have or do not have a nucleus, are going to have a plasma membrane that surrounds the outside of the cell. A synonym or common term that's used for that plasma membrane is cell membrane. So if you see plasma membrane, think of it as a synonym for the term cell membrane. Both are equally correct. On the inside of the cell, we have a cytoplasm. The cytoplasm is a liquidy substance that contains both liquids and structures of the cells. Those structures on the inside of the cell, generally speaking, are referred to as organelles. A little, LA, meaning little, and then organ. So an organelle is a little organ inside of the cell. And both prokaryotic and eukaryotic cells also have DNA. Now, prokaryotic cells do not have the DNA and nucleus, but they still have DNA. Eukaryotic cells do have DNA. Their DNA is stored in a nucleus. So let's look at a eukaryotic cell. Here is an electron micrograph of a eukaryotic cell at 10,000 times total magnification, and you can probably see, if you get really close to the video monitor, see lots of little structures on the inside of this eukaryotic cell. The eukaryotic cell has many membrane-bound organelles on the inside, including a very large central nucleus. So here's a cartoon figure of the eukaryotic cell, and you can see many different organelles. The nice thing about this cartoon figure is that it's easier to see the structures in it compared to these electron micrographs. If we look at our eukaryotic cells, it's believed that a eukaryotic cell started off as a precursor prokaryotic cell, and that this precursor prokaryotic cell had some invaginations form in the membrane, and those invaginations eventually became the membrane-bound organelles, and then that prokaryotic cell phagocytized or swallowed aerobic bacteria, or bacteria that needed some oxygen, and then those bacteria had a symbiotic relationship inside of the prokaryotic cell and turned into nuclei, and that gave us an animal cell. And then also, to get plant cells, some photosynthetic bacteria were also phagocytized that acted as the chloroplast to give us our plant cells. So, concept check, class. What kind of cell do we have in the picture shown? This cell has a mitochondria, it has a ribosome, it has a rough ER, so it has lots of membrane-bound organelles, and it has a nucleus in the center containing its DNA. So is that going to be a eukaryotic cell, a bacterial cell, an archaeal cell, or I cannot tell based on the picture. Go ahead, pause the video, and flip back in your notes, flip back in your textbook, get me an answer. 1, 2, 3, 4, 5. The correct answer is a eukaryotic cell. It's a true kernel, eu meaning true, kary meaning kernel. It's a true kernel in that it absolutely does have a nucleus in the center. So it is a eukaryotic cell. Let's continue on with the discussion on the plasma membrane. Remember, the plasma membrane is also known as the cell membrane. It's on the outside of the cell, and so it separates the outside environment from the inside environment of the cell. Our plasma membrane is made of a phospholipid bilayer. This plasma membrane has many proteins embedded in it. These proteins are constantly jiggling around within the plasma membrane. Within the plasma membrane, there's also cholesterol. Yes, cholesterol is a necessary part of our plasma membrane. Many people think that cholesterol is bad for you. What is a better answer is that excessive cholesterol is bad for us. We need small amounts of cholesterol in our body to maintain our cell membranes and to make our steroid-based hormones. The cholesterol within the cell membrane helps the cell membrane to have structural support, to be more viscous, particularly in higher temperature environments. Our cell membrane also has carbohydrates that are embedded on both proteins and lipids. These carbohydrates act as an ID tag or a label. And then finally, our plasma membrane of the cell is selectively permeable. It lets some things go in and out, and other things it blocks from going in and out. You can think of it as a colander. It lets some things pass, but not all things pass. Here's a figure of our cell membrane. We can see the most common molecule is this little blue thing with the two squiggly lines coming off of it. That is the phospholipid. So phospholipids make up the majority of the cell membrane or the plasma membrane. On the outside surface, the extracellular surface, we have carbohydrates stuck to proteins and carbohydrates stuck to lipids. So glycoproteins are glycogens or carbs stuck to a protein. A glycolipid is a carbohydrate stuck to a phospholipid. The glycoproteins and the glycolipids serve to identify the cells and will form a series of structures on the surface known as the extracellular matrix, the ECM. The extracellular matrix helps to embed the cell within a tissue. Inside of the cell, we have filaments that make up the cell skeleton or the cytoskeleton. We can also see small dark purple cholesterol molecules embedded in the cell membrane that help give structural integrity to that cell membrane. And then finally we have big red protein molecules. Some of those protein molecules will be tubes going through the cell membrane that let some things in and out. Other protein molecules are going to go through the plasma membrane and then have a peripheral protein attached to the inside so that when a signal binds to the outside, this peripheral protein on the inside can be released to transmit that signal. Let's spend some more time talking about selectively permeable. What exactly do I mean when I say selectively permeable? I mean that the cell membrane will let some things go in and out while other things cannot pass through the cell membrane. So if we look at highly charged particles cannot exit the cell membrane, through the cell membrane, or enter through the cell membrane. They need to have a special carrier protein. Water molecules are a good example. Water molecules are fairly charged, or have a significant charge to them. And they can only go in and out of the cell membrane through special protein tubes. In this case, it's an aquaporin, quite literally a water hole. Non-charged or non-polar molecules can go straight through the cell membrane. These phospholipid tails, or these fatty acid tails of the phospholipids, are hydrophobic. They are non-charged and they do not like being exposed to water. The hydrophobic properties of these fatty acid tails mean that these water molecules can't go through the fatty acid tails. They need to go through the carrier protein. But a non-charged, a non-polar molecule can go straight through. Very large molecules, like a large protein or a large carbohydrate, are too big to go through the cell membrane, and they typically are going to need to be phagocytized or exocytized to pass through the cell membrane. So, concept check, class. How do things move across the plasma membrane? Do they move through diffusion, osmosis, facilitated transport? Oh, actually, this is not a concept check. My bad. So let's talk about how things can move across the plasma membrane. There are 5 different ways. I was wondering, oh my God, I didn't talk about those yet. So there are 5 different ways we can move things across the plasma membrane. They are diffusion, osmosis, facilitated transport, active transport, and and exocytosis. So let's first talk about diffusion osmosis. Diffusion is just the movement of a molecule. It is completely random. It doesn't require any energy. It happens spontaneously. And molecules will move from an area of high concentration to an area of low concentration. When we look at these molecules, generally speaking, they're going to be small molecules. They're going to diffuse through an aqueous solution. A good example is making tea. The tea molecules can diffuse from the tea bag through the entire glass of water to fill the glass of warm water with tea molecules. Osmosis is a very special kind of diffusion. It's the diffusion of water molecules across a semi-permeable membrane. And we need both kinds, the diffusion of regular generic chemicals and the diffusion of water to help keep our cells alive. So if we look at diffusion and osmosis, here's a good example of both of them taking place. We have lots of water on one side of the plasma membrane and a little bit of water on the other side of the plasma membrane. So the water is going to move from the right side to the left side. We have bigger arrows pointing to the left. And now the water has equalized across the membrane. We also have random chemical particles, and we have high concentrations of the chemical on the outside, lower concentrations of the chemical on the inside. And our chemical particles also go from an area of high concentration to an area of low concentration, so they move from the right to the left, and now the chemicals has diffused, the water has osmosed, and now the concentrations on both sides are equal to each other, so we've reached equilibrium conditions because there's no more net movement of the molecules from one side of the membrane to the other side of the membrane. We have some special terms that can measure the solution concentrations inside and outside of the cell, and those are the tonicity terms. If a solution is isotonic, there's an equal amount of chemical inside of the cell and outside of the cell. And this would be an example of an isotonic environment. During that isotonic environment, there's no net movement because the concentrations are equal on both sides of the membrane. So in an isotonic environment, the cell does not swell up and it does not shrink, it stays the same size. Generally speaking, when you give a patient an intravenous solution, you want that intravenous solution to be isotonic to the patient's blood plasma. We also have hypotonic. Hypotonic solutions have a less dissolved solute outside of the cell compared to the inside of the cell. And because there's lower concentrations of chemicals outside of the cell, we are going to have lots of water diffuse into the cell, and as that water diffuses into the cells start to swell up. So here we can see nice, happy red blood cells in an isotonic solution with their classic dimple in them. These are red blood cells in a hypotonic solution. They are swelling up and losing their little dimple. We also have isotonic solutions, hypertonic solutions. In hypertonic solutions, we are going to have a lower or a high concentration of chemical outside of the cell compared to the inside of the cell. That high concentration of solute outside of the cell causes water to osmose out of the cells and the cells to be reduced in volume. This leads to the crenation of the cell, and crenation is a technical term that means shriveling of the cell. So you can see the water leaving the cell, and the red blood cells start to shrivel up. They get these wobbly membranes. We also have facilitated diffusion and active transport. Facilitated diffusion is going to be the movement of a molecule across the plasma membrane, across that cell membrane, with a transport protein, with a protein carrier. So here we can see a tubular protein, and that tubular protein can move glucose molecules from areas of high concentration to areas of low concentration, from the outside to the inside of the cell. Glucose by itself can't pass through that phospholipid bilayer that makes up the cell membrane, but glucose can go through the carrier protein. So it needs something to help it move through the membrane. It needs something to facilitate its transport. Facilitated transport is going to be done via a protein carrier. And I should also mention, facilitated transport does not require energy. It always goes from areas of high concentration to low concentration. It's spontaneous in nature. Active transport, on the other hand, does require energy. Remember, cellular energy, or the energy currency of the cell, is adenosine triphosphate, ATP. And we can use this energy to move the chemicals from areas of high concentration, from areas of low concentration to areas of high concentration. So we see here that we have hardly any sodium in the cell and lots of sodium, lots of NAs outside of the cell. We're going to take the sodium from the inside of the cell and pump it out of the cell. So it'll go from an area of low concentration to an area of high concentration. And this does not happen spontaneously. It requires the use of energy. Active transport is when we use a protein pump to pump a chemical against its concentration gradient from an area of low to high concentration. And then finally we have endocytosis and exocytosis. Endocytosis is when the plasma membrane pinches and brings a large amount of molecules into the cell. There are three kinds of endocytosis that forms from that pinching or invagination of the plasma membrane. We have phagocytosis. Phago is a word meaning to eat. Phagocytosis is when the plasma membrane pinches in and brings in a solid substance to the cell. Penocytosis means to drink, cell drinking. Peno is a prefix meaning drink. During penocytosis, the cell membrane invaginates and brings in liquid. And then finally, we have receptor-mediated endocytosis. During receptor-mediated endocytosis, the cell membrane invaginates and brings in very specific chemical compounds that are bound to receptor proteins in the plasma membrane, and then brings those very specific chemical compounds into the cell. So endocytosis is bringing that substance into the cell. There are three kinds of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. And then finally, we have exocytosis, where we bring something out of the cell. Exo meaning out of cell. During exocytosis, we have the opposite take place. Instead of having the cell membrane invaginate and turn into a vesicle, we'll have a vesicle bind to the cell membrane and then release its chemical substances to the outside environment. So, concept check, class. Whenever a chemical is being moved against its concentration gradient, is energy being used? Will the cell lyse? Will it happen spontaneously? Or more than one of the above? Go ahead and rewind the video, or pause it and check your notes, or check your textbook and get me an answer. 5, 4, 3, 2, 1. The correct answer is energy will be used. Whenever you move that chemical against the concentration gradient, energy will be used. If you have any questions about the content of this video, please feel free to post them in the class discussion board, shoot me an email, or talk to me on campus. Happy studies!